A new type of coronavirus has emerged from the city of Wuhan in China. Within weeks, it has spread to several countries and has killed multiple people. Corona refers to the virus's distinctive shape. They are a large group common among mammals and birds. But in rare cases like this one, they can spread to humans, a process known as zoonosis. This new virus is the 7th known human coronavirus, and it affects the respiratory system. Some coronaviruses have mild symptoms, however others are much more dangerous. SARS and MERS are both coronaviruses.
The disease spreads through small droplets that are expelled from the nose or mouth when a person with COVID-19 coughs or exhales. Therefore, standing close to someone who is infected can put you at risk. These droplets can land on your hand and be transmitted through something as simple as a handshake, if afterwards you touch your eyes, nose, or mouth, the so-called T-zone. The virus is known to survive on different types of surfaces, so touching these contaminated surfaces and then touching your T-zone brings a high risk of infection.
The large S glycoproteins are used by the virus to gain entry to human cells. They most likely attach to angiotensin-converting enzyme 2 receptors on the cell membrane, allowing the virus entry. The exact mechanism for this is not known. Most likely, as shown here, the human cell ingests the virus in a process known as endocytosis. Once inside the cytoplasm, the endosome opens to reveal the virus's genetic material, a single-stranded RNA. The virus hijacks the cell's machinery to replicate the RNA in N proteins and uses the endoplasmic reticulum to form its M protein outer layer and the all-important S protein. After replication, the virus is carried by the Golgi bodies out of the cell in a process known as exocytosis, so that it can infect other cells. Meanwhile, the stress of viral production on the endoplasmic reticulum eventually leads to apoptosis, or cell death.
When the coronavirus attaches to a cell, it begins to replicate within the cell. The body's immune system attempts to destroy the virus, which results in an inflammatory response that causes fluid accumulation in the lungs. As the lungs fill with fluid, the body's available oxygen decreases, which can lead to organ injury and death. The risk of serious complications increases if a person is elderly or has associated medical conditions.
It's a specimen collection tube with 2 swabs. I'm just trying to give you a demo. How does the swabs break after taking the specimen collection? Okay. This is a tube. Let us consider that this swab has already collected the specimen. Just place it like this. Use your thumb and index finger. It breaks. See, it simply breaks. Considering this also has collected the specimen from nasal throat, I'm placing it over here like this, bending it using my index finger, it simply breaks. Now it has 2 swabs within this. We're just closing it up like this.
So this is the second case for which the sampling will be taken by Dr. Ravikant again. So he's getting ready. So he's almost ready. Sanitizing the hands with sanitizer. The patient is also ready. It has been sprayed another time. So doctor is inserting the hands again through the glass plane. So he has taken the swab, removing it, just taking it out, and that is the swab with which the sample is taken from the throat. So that's it. That is the time when patient is prone to cough, and that's when the glass pane helps protecting the doctor. So it's, and that is the, he's opening the, so it is introduced into the bottle having the transport medium along with the swab, and it's being kept into the container having ice packs. So the spraying will be done again to the hands, and the person is protected with PPE of operation theatre. So hands are removed now. So that is the total procedure. Thank you.
To begin the reaction, the temperature is raised to 95 degrees. At this temperature, the double-stranded DNA is separated into single strands. Then, 3 major steps are repeated during each cycle. First, denaturation to obtain single DNA strands. Second, annealing. The temperature is then lowered to approximately 60 degrees. This allows the primers and the fluorescent probe to bind to your target sequence of interest. And third, extension. Between 60 and 72 degrees, the polymerase extends the primers and cleaves the probe, which generates an increase in the fluorescence signal proportional to the amount of amplified product. After one cycle of PCR, there are twice copies of the target DNA. These steps are repeated through around 45 cycles. When the starting material is RNA, one first additional cycle of reverse transcription is required. The real-time PCR reaction requires the following components: Taq DNA polymerase, reverse transcriptase, primers, probe, DN-TPS, MgCl2, magnesium chloride, buffer, stabilizers, internal control. BeAssure real-time PCR detection kit contains the following components: well strips with all the previous reactives in a stabilized format, positive and negative control, rehydration buffer to reconstitute the stabilized product, water RNA SE DNA SE free, optical caps for sealing wells during thermal cycling. Molecular diagnosis unit consists of 4 areas: nucleic acids extraction, pre-PCR, setup, PCR, amplification and results interpretation. The first area aims to process potentially infectious samples and subsequent extraction and purification of nucleic acids with the RNA-DNA BeAssure extraction kit. We recommend to dilute stool samples before extraction and use the supernatant to perform DNA-RNA extraction. Homogenize the fecal sample, collect a pea-size and transfer to a 2 ml vial with 200 microliter of RNase DNase free water, PBS or physiological serum. Vortex the sample and centrifuge. Samples are lysed at elevated temperature in the presence of lysis buffer and proteinase K. Transfer the supernatant to the 2.0 ml collection tube. Then add lysis buffer and proteinase K and vortex vigorously. Incubate 10 minutes at 65 degrees and then 10 minutes at 95 degrees. After optimal binding conditions are adjusted by the addition of isopropanol, the lysate will be applied onto the mini-spin column and DNA-RNA bonds to the surface of the filter membrane. Add binding buffer and mix by pipetting or vortexing. Incubate the sample at room temperature and transfer the lysate onto the column mini-spin column set. Centrifuge and discard both the filtrate and the RTA collection tube. Then place the column mini-spin column into a new RTA collection tube. Contaminations and enzyme inhibitors are efficiently removed during the following wash steps, while the nucleic acids remain bound to the membrane of the mini-spin column. Add wash buffer 1, centrifuge and discard. Add wash buffer 2 and centrifuge. Repeat this washing step once. Centrifuge to remove ethanol residues and discard the filtrate and the RTA collection tube. The nucleic acids are finally eluted from the membrane using elution buffer. Place the column mini-spin column in the sterile 1.5 ml collection tube. Add elution buffer preheated at 65 degrees. Incubate at room temperature and centrifuge. Discard the column mini-spin column. Store the DNA-RNA sample at 4 degrees for immediate use or preserve from -20 to -80 degrees for longer periods. The sample is ready for the next area. Take the BeAssure Pathogen Real-Time PCR Detection Kit and separate the required number of wells, including samples and 2 reserved wells, for the positive and negative controls that must always be included in each assay. If necessary, cut the required number of wells with the scissors and remove the protective aluminium seal. Reconstitute each one of the wells with 15 microliters of the blue vial rehydration buffer. Finally, add 5 microliters of the purple vial negative control in the reserved well for negative control. After this process, move the rehydrated well to the next area. 2 independent processes are made in the third area. First, the positive control rehydration. ViaSure Pathogen Real-Time PCR Detection Kit contains a positive control that must be rehydrated before using this assay. It is recommended to open and manipulate before starting with the assay. Reconstitute the red vial positive control with 100 microliters of the white vial RNase, DNase free water and mix with the help of the vortex. Store at -20 degrees after resuspension and divide in aliquots to minimize the freeze-thaw cycles. The second process is the addition of the nucleic acids and positive control to the rehydrated wells. Add 5 microliters of extracted DNA, RNA from each sample in the rehydrated wells, with the exception of the 2 well reserved for the positive and negative control. Add 5 microliters of reconstituted red vial positive control in the reserved well for positive control. To finish, divide the required optical caps number and close the wells. After this process, move the plate to the real-time PCR equipment. Place the plate in the thermocycler, program the thermocycler and start protocol. When finished, analyze the results with the software installed in the equipment. ViaSure provides maximum flexibility and compatibility with leading open system thermal cyclers and the latest technology for the stabilization of products.
Hidden in the natural world around us are powerful biological tools whose potential scientists are just beginning to unlock. For billions of years, bacteria have been under attack from viruses. But bacteria developed clever defense mechanisms to remember these viruses and fight back. Recently, scientists harnessed one of these natural tools called CRISPR-Cas9 and created a powerful gene editing technology with the potential to treat human disease. But Cas9 isn't the only CRISPR tool with applications for human health. A team of scientists found that a cousin of Cas9 called Cas13 can be harnessed to actually detect human disease. Here's how Cas13 works in its natural environment. First, it hunts for viral RNA using an RNA guide. When it finds its viral target, Cas13 becomes activated. In some circumstances, it cuts any RNA it encounters, a process termed collateral cleavage. It is this mechanism that scientists at the McGovern Institute, Broad Institute, and Harvard University harnessed to create Sherlock, a highly sensitive tool to detect infectious diseases in humans. Here's how the Sherlock detection system works. First, researchers take a sample from a patient with a possible viral infection, like the flu. Then, they amplify the levels of RNA in it and add reporters that are sensitive to Cas13. Then, an engineered CRISPR-Cas13 is added to the sample. This Cas13 is programmed with a guide RNA that is designed to find only virus RNA and bind to it. When this happens, Cas13 activates its cleaving mechanism and begins randomly slicing nearby RNA, including the reporters. Since each end of the reporter carries a different label, Cas13 separates these two signatures, creating a unique signal within the sample. The sample is then applied to a commercial flow detection system. If the sample is negative for flu, the reporter remains intact and collects at the first detection line. If the sample is positive for flu, it collects at a different location, making a diagnosis easy to spot. So easy, in fact, Sherlock can be used in the field to detect emerging infectious diseases and outbreaks, like Ebola virus.
Washing your hands with soap is possibly the simplest way to flush pathogens down the drain. Soap molecules can interact with pathogens, such as coronavirus, leading to their destruction. Each soap molecule has two parts. The head is hydrophilic, which means it likes to interact with water, while the lipid tail is hydrophobic, which means it wants to get away from water to find similar water-avoiding particles. This is the basis of how soap works. When soap molecules encounter lipid particles on our skin, the tails aggregate around them to form spherical structures called micelles, creating a water-free environment. Similar things happen when they encounter enveloped viruses such as coronavirus. Envelope viruses carry their genome and supporting proteins inside a lipid bilayer membrane, with the proteins necessary for infection embedded in the membrane. In cells and viruses, these lipids are packed neatly into two sheets with the lipid tails facing inward. The lipid ends of the soap molecules are attracted to the lipids in the membrane. They take advantage of the presence of membrane proteins which can perturb the neatly organized bilayer to insert themselves into the viral membrane. If there is only a small amount of soap, the soap molecules only loosen the membrane. But with more soap, they begin to create micelles around membrane lipids. They are also attracted to hydrophobic amino acids in the membrane proteins, extracting them from the membrane. The structural integrity of a virus is essential for infection. So, washing your hands with soap protects you better from infection than washing with only water.